Hi everyone. I warmly welcome you to this forum, this webinar this evening. My name is Olorundare Lufunke. My name is Olorundare Lufumilola. I'm a Nigerian. I have a master's in medical microbiology from the University of Jos, and also I have associate in medical laboratory science in medical microbiology also, and also have a fellows in hematology, a fellow in hematology in medical, in hematology from the Federal College of Veterinary and Medical Laboratory Science, NVRI, we call it JOS, Nigeria, Plateau State. Presently, I'm going through my PhD, students carrying out a research on Salmonella phage. I'm a Nigerian, and Nigeria is situated in Africa. Africa is a place to be. Sorry, let me share my slide. I'm finding it a little challenging. It's not coming up.
Not yet.
Not yet.
What happens when you click share screen? What do you see?
Oh, okay. All right. So I'll just move to the second slide. Nigeria is a lovely place to be. Nigeria is situated in Africa, West Africa precisely. It's a lovely place where God has really blessed us in Nigeria with a number of ethnic groups, from 250 to about 300 ethnic groups with different languages, diversities of ethnic groups in Nigeria. It's a place to be. We have good weather, good cultural and traditional practices. Our flag is noted with green, white, green, which signifies wealth, peace, and unity. We have about 36 states in Nigeria with the Federal Capital Territory, which is in Abuja, the north central of Nigeria. It's a lovely place to visit, and I will want us one day to propose to visit Nigeria. Even when you have a Phage forum, a physical meeting, we have lovely sites, and that will take us to tourist attractive sites in Nigeria. We have a number of them, a number of tourist attractive sites in Nigeria, but just a few that will be shared with us, a few sites. I will just start from where I reside. I reside in Jos, North Central Nigeria, Plateau State. And we have what is known as the Sherry Hills. Sherry Hills is noted for these undulating rocks and very, very high, which also attracts a lot of tourists. It attracts a number of people, those that have ICAS dreams, that they long to come to Sherry Hills. It's a lovely place to be. We have a serene environment there. We have halls, conference halls where meetings could be held. It is known as one of the highest peak, the third highest in Nigeria. It's very, very high, of about 1,829 meters high. That's about 6,000 feet high. And so many people, it has really attracted a lot of tourists in the country. The second one is the Ikogosi Warm Spring in Ekiti, Western Nigeria. Surprisingly is that this spring has two ways. There is another one inlet that comes with cold water. Another one comes with warm water of about 70 degrees. But this water has a confluence where they meet, but the water does not mix up at all. One side is warm, the other side is cold. It is so unique that it attracts a lot of tourists. We have so many guest inns around there where you can lodge in and have a very good site for tourists that come around the world. Another one is the Olumo Rock. Olumo Rock is situated in the southwest also of Nigeria. It is a rock that has attracted a lot of tourists. In those days when we have ethnic clashes, they have caves where people hide in. And so it has attracted, you can see a number of tourists that came there around to check things that are being done there, a lot of caves, a lot of hideouts, where people can find refuge. It is a lovely place to be. Whenever you come to Nigeria, it's a place that you can visit. We also have the Yankari Game Reserve. Yankari Game Reserve, we call it Yankari National Park, Bauchi, a lovely place to be. I can say it again, a lovely place to be. We have wildlife there. We have pools, serene environments. There you can see your elephants. Wow, beautiful. We have the zebras, we have lions. We have a number of animals there. Tourists, in fact, it's a great tourist site. It is situated in the northern part of Nigeria. We call it Bauchi State, northern part of Nigeria. It has attracted a lot of tourists. In fact, so many lovely sites that can be found in the Yankari National Park. Another one we have here is on the Cross River, which is the south part of Nigeria. We call it Agbokin Waterfall in Cross River, a lovely waterfall. This waterfall it falls with great force, it's a lovely sight for people around there. It's the waterfall, we call it, it has 7 channels that comes in. Water comes in there through 7 channels. So we normally call it 7-faced wonder. 7-faced wonder. It's a lovely, also a lovely tourist site. It shares border with our neighboring country, the Cameroon. So when Cameroonians want to visit, it is close by. It's also a good tourist site for them to visit. Another tourist attractive site here is sorry, I'm trying to share the second slide. Yeah, the other one, we have the Obudu Mountain Resort. We call it Obudu Mountain Resort, Cattle Ranch. It is a lovely place. We have waterfalls there. We have cable transport. We have so many serene sites where so many people around the world could visit. We have also the Mambila Plateau. It's located in Taraba State, the northern part. The Mambila Plateau is a very high plateau, the highest mountain in Nigeria. It is a lovely site. The plateau is a lovely site. So many serene environments along the Mambila Plateau of Nigeria. We also have the Kanji Dam. The Kanji Dam is in the state called Niger State of Nigeria. This Kanji Dam, we cannot do anything in Nigeria without Kanji Dam, because this is the source of our hydro power, hydroelectricity power. It is from this Kanji Dam, a number of turbines are there which generate the power that is used across the country, Nigeria. So you cannot talk about a dam in Nigeria without mentioning the Kanji Dam, because without the Kanji Dam, we will not have a power supply in the country. Another one is
Olufem, thank you so much for the introduction. However, because the speaker was just in for a short time, I think we keep this to the end so that at least we can enjoy the meals towards, at least people will have some food through, well, we all are doing things through virtual. So, but I would really like to introduce Arshnee because she's running out of time, and I'm so sorry for interrupting.
Yeah.
Yes, so as Arshnee is bringing up her slides, and thank you so much for showing the tourist attractions and everything. So Arshnee, would you want to bring up your slides? So I want to introduce Arshnee, as Arshnee leads the CGIAR AMR Hub that was launched in 2019 with the aims to support efforts to mitigate the risks of agricultural associated antimicrobial resistance in low and middle income countries, working within the One Health framework. She is a microbiologist with a PhD in antimicrobial resistance and zoonosis from the University of Copenhagen, Denmark. She joined the International Livestock Research Institute from the Department of Veterinary and Animal Sciences in University of Copenhagen, Denmark, where she has been an associate professor in AMR research group, and heads the PhD graduate program in molecular bacteriology and infection, and was responsible for the NGS service at the section. She was heavily involved in Antibiophage, an EU-funded project to develop and test a bacteriophage cocktail to treat colibacillosis in poultry. At the University of Copenhagen, she was part of a team that developed 8 cocktails, which in December 2019 tested 2 of their in vivo efficacy. She also has vast knowledge on other stubborn bacteria that affect different animals and how phages may play a part in tackling AMR in her experience. So Arshnee, glad to have you as a speaker today, and please feel free and start whenever you can.
Thank you very much, Angela. You see me? I'm just going to switch off my video now because of my bandwidth. Sometimes here in Kenya, the power goes off and all kinds of things. So I'm hoping that that does not happen today. So let me just switch off my video. So firstly, I want to say thank you very much to Angela, and she's been asking me to, for a while now, to come here and talk to you about my work with phages. And I've been reluctant because I don't consider myself a phage researcher. I feel like I'm an imposter in this field, and so have been a little hesitant to masquerade as somebody that knows anything about phages. But she persisted, and now I'm here to tell you a bit about what I've been doing the last 10 years in this field. It's basically something I stumbled upon. As I told her, I do not consider myself an expert in the field. I merely see phages as a tool to achieve what I am working on, and my life's work is around antimicrobial resistance and addressing that. And as she said, yes, so I'm here in Nairobi, Kenya. I'm based here, and that's my email address at the bottom there. So if you have any questions, you're welcome to drop me an email after this. Let's get started. So I'm South African, so I'm from the continent, but I moved to Denmark many years ago to do my PhD there. I'm still associated or affiliated with the University of Copenhagen. I have my professorship there, but now I'm here in Kenya. And as Angela said, I joined in 2019 to lead this newly established antimicrobial resistance hub, which is a CG-wide, with the goal to sort of bring or address the agricultural AMR risks in low and middle income settings. I also lead the AMR team here at ILRI, who is the host of the hub. And the hub really is an interesting platform, and I call it a platform because that's really what it is. It's a convening platform for all of the research that we're doing within the CG with our external partners to address various aspects of antimicrobial resistance, particularly focusing on agriculture. And agriculture being livestock, crop production, as well as aquaculture, and all the things that connect that in terms of wastewater, and food and things like that. So it's AMR is an incredibly complex problem, and so it needs this multi-sectorial, multi-level approach. But more important, it needs a focus that is very much low and middle income country driven to look for sustainable interventions. And that's what we're doing here. And this is the hub. And if you want more information about the hub, please check out our website. But I didn't come here to talk to you about that. But phages are one of the red pillar, which is the interventions, and potentially play a role in that. So I'm going to tell you the journey of my life. So starting way back in 2005, when I left South Africa to go to Denmark, which is almost 10,000 kilometers away, and speak a very funny language in a very small country, to do my PhD. And on the left is what my thesis looked like. I graduated in 2008, with a PhD focusing on resistant bacteria that was potentially transferring from animals to humans. And it's a picture of a farmer, and it's got pig, horses, because that's what I was working on. And I was also working with veterinarians and dogs. And at the bottom right, the organisms that I was focusing on was methicillin-resistant Staph aureus, and this was in pigs, because when I joined Denmark at the time, MRSA in pigs was starting and is now, widely, so much research has gone on within this area. We were also looking at MRSA in dogs, And at the time, dogs were seen as a reservoir for MRSA in humans. And you should know in Denmark has a search and destroy policy around MRSA. This is in part related to the healthcare system. It's a free healthcare system. So to protect the healthcare system, a number of pathogens are on these control search and destroy policies, similar to what they have in the Netherlands. I also worked on extended spectrum beta-lactamases, namely in E. coli in pigs, and then also on methicillin-resistant staphylococci in horses. And really, my favorite organism to work with is staphylococci, and in particular Staphylococcus aureus. And that's where my love started. And that's how I got into phages, actually. So it was really by coincidence. As I said, I was working on these staphylococci, and a colleague of mine from the UK contacted me because she had found these, not Staph aureus, but a different kind of staphylococci in dogs that was suddenly very resistant. And so this is Staphylococcus pseudointermedius, which is very much like Staph aureus is in humans. This is the Staph aureus or type of Staph aureus in inverted commas in dogs. So it's a commensal that is found on the skin and mucosa. But it's also a very common, it's also an opportunistic pathogen. Given the right conditions, being a skin commensal, in the right conditions, it can cause a number of skin, and it's mainly associated with skin and ear infections. And typically,
Arshnee,
yes?
are we still on whom am I, the slide?
No, I've moved on.
Yeah, apparently there's no syncing between the slides.
Oh,
maybe you can reshare
okay.
again your screen.
Let me
and
stop
then,
sharing.
Yeah, and then reshare again, yes.
Oh, I had the same problem earlier. Let's go, let's see. Where is my?
Yeah, it's like slides are not changing.
Okay, I need to fix that at some point. So let's go there. And you see this, right?
Yes.
Okay.
Arshnee has started screen sharing.
Yes, and do you see this slide?
Yes, as you're moving, yeah.
Okay. So I'm not going to go into the presentation mode because I think that's what happens normally with my computer, is that it tends to crash.
Okay.
So I'm going to try to make this bigger, and you tell me if it's big enough and
Arshnee,
you can see that.
I don't see your slide. I see a black screen. Am I the only one?
It's saying Arshnee has started screen sharing.
Yeah, but that's all it says.
Yeah, that's what, yeah.
Okay, you know what I'm going to do? I'm going to send it to you, Angela, my slides.
All right, okay.
And then you see if you can share it on your side.
Yes, and then you'll just be telling me next slide.
Yes, I'm going to put it
Cool.
in our Teams, Angela.
All right, on
Let's
it.
see if that works. I'm so sorry, it's so annoying.
Yeah, we've had this at another event. It's weird how it happens.
It's just loading, Angela, 1 second. It's a big file because there's so many pictures. Let me also put it on a flash drive just in case in the meantime. If it doesn't work. Where's my flash drive? Of course, when you need it. Okay, Angela, can you race to my office and get it? It's under today's date. It's called Phages. Yeah. Okay, so Angela's just raced down, gotten it, and is racing back.
So nice to have in-person things.
Yes.
Forgot about that possibility.
Angela, it's also in your Teams if you have difficulties opening my flash drive, because that can happen.
Everyone can go get a coffee and come back. Should be a couple minutes.
I hope this will work. So I have the slide, and maybe Arshnee can tell me where she was.
No worries.
Yep.
I don't see
I'll
it
just wait.
It's weird, Jessica. I thought we did a test and you could see it. So
Yeah, we did. Yeah.
okay.
Yeah.
Yes, that's where we are. So the one on my left is my PhD thesis, the cover of my thesis. And as I said, that was the work I was doing. So next slide. Yes. So this was a friend of mine that was working in the UK, Annette Luffler, she contacted me because they had been isolating some Staphylococcus pseudointermedius, which is this commensal that is found on the skin and mucosa of dogs. And basically, it was not, these animals were not responding to the normal treatment for a skin infection, which is normally or typically cephalosporins, which is a beta-lactam. So they had then saved a number of these isolates from it. I think it was 12 different dogs in Germany, and sent it to me to try to investigate why were these isolates now resistant. And this was despite being on long-term therapy. So when you have a skin infection or pyoderma, depending on the degree or the severity of the infection, treatment is either topical or it's systemic. And when it's systemic, it tends to be very, very long-term systemic therapy. And in this case, these dogs were getting it sometimes between 4 to 6 weeks of treatment with different antibiotics. And so what we discovered that the mechanism of resistance was very similar to that is found in MRSA, which is MEK-A mediated. But more interestingly, and subsequent work that we did showed that it's, at least in Europe, you had this clonal expansion of a dominant clone that was had the sequence type 71. Next slide, please. But more and more people would then, after we had published our paper which came out in 2007, more and more people started describing this methicillin resistant strain and the resistant profiles to that. And so in red, you have resistant, which is the methicillin resistant, and in green would be your methicillin susceptible strains. Basically, what I wanted to highlight is that a number of these isolates were multi-drug resistance, often resistant to very many different classes, and sometimes leaving very few or none or no treatment options for these for these animals. Next slide, please. And so what was left really were that to which those isolates were susceptible to are drugs or antibiotics that you typically would not use for treatment, either because they're not available in the country, or because of toxicity issues with long-term usage, or because if they're used as a monotherapy, you will have resistance development. And there were some of the strains were susceptible to drugs that were not licensed for use. And those are typically the drugs that would be critically important in humans to treat gram-positive infections in humans, like linezolid and vancomycin. And really the debate at the time was, or still is going, should you be using drugs that are not licensed for use in animals, and also drugs that are considered last resort or critically important in humans. And so you had this issue with euthanasia, or sort of dogs being put down because we're not having anything left to treat them. Next slide, please. And that's really where we started with, or how my journey into phages started, was that, well, if we don't have antibiotics to treat these dogs and we don't have other things that we can do, why don't we look at phages as a possibility for that? And so I've put the links to, or at least I've put the sort of the papers that we have published on the various work. So this was a work that was done together with a master's student back at the University of Copenhagen. And essentially we got in contact with a guy that we knew was working in food microbiology. His name is Finn Vogesen. And Finn was, and his team had been looking at phages in Lactobacilli and Leuconostoc and it was related to the food industry. And so he had a vast experience. And so we went to him to say that this is what we wanted to do. And then he became a very good friend of mine, and still is a good friend of mine, and has helped me navigate this field. And so we, you know, if you're into bacteria and into phages, you work a lot with poop and sewage and all of that. So I'm that girl as well. So we isolated phages from dog feces. So if we had a dog, if we were in a park and an owner and the dog made a poop and he was about to pick it up and throw it in the bin, my student would be running after him in order to get that. So yeah, we were those girls in the mornings. And so we took this to the lab, and we managed to isolate phages in all of the samples, but the titers were very different between the different samples. And so we chose to pick those samples that gave very high titers, and then pick one phage from that particular sample, and then ended up with 7 phages. And so we did some microscopy, electron microscopy, and that's the figure on the right side, that beautiful picture, and basically showing that in the morphology of the phages are those that is very reminiscent of phages belonging to the Cipheridae. And then at the end, we picked only 4 phages to move forward because they all look the same, and thinking that, well, if they look the same, then they're likely to be the same. And so we did, the very first thing we did is like the host range, and we saw that all of the resistant strains that we tested the 4 phages against, they produced clear plaques. And on the far right, you have this almost like a traffic light system. Those are the methicillin susceptible strains that we tested. And basically showing that we could get these clear plaques. Those are the ones in red, but most of the time you had very turbid plaques or turbid plus clear plaques. And that was really good for us because we wanted phages that would be specific for the resistant strain, but left the susceptible strains alone because we wanted them to be part of the microflora. And then we did a little bit of genomics. We sequenced our phages. They were about 40 kb, and they were very similar to each other as we suspected. But then came the sad news, is that all of the phages had this lysogeny module. So that being temperate phages, and obviously are not a good option for therapy. Next slide, please. And this is about as geeky as I'm going to get when it comes to talking about phages. But the interesting thing with our phages was that they behave very much like lytic phages. They consistently produce these clear plaques on strains that we knew had prophages because we had also sequenced our bacterial strains. And so we were quite curious to look into this. So when we looked at the sequence of our phages, and we looked at prophages that were in the bacteria, you could see that they were very, very similar. And really the only difference was mostly in the lysogeny module, which is the yellow module on the far right. And so we did a number of experiments trying to propagate phages on different strains and then sequencing them to see what was sort of left over. And basically what we could see from the sequence data was that we were seeing recombination between the temperate phages and prophages that then would allow, because they would then recombine and swap the lysogeny modules, we were then able to infect phage strains that had almost identical prophages. And this is not novel in terms of, it's been seen and known to happen in E. coli and in lactococci as well. But basically the story ended with, well, we got a bunch of temperate phages. This is not possible to take forward for treatment, but there are ways in which you can knock out the lysogeny module naturally, because if we genetically modify the phage, that would open up another can of worms that we didn't want to really go into, because we were ultimately looking for tools to be used for treatment. So next slide, please. So while this was going on, I was going to different veterinary association meetings and telling them about the work that we were doing, because we had a very close contact with small animal veterinarians with other projects. And then I got a phone call from a vet who had heard me talk about the work that we were doing with staph Pseudointermedius, and contacted me about a case of a dog that had recurrent ear infection. So ear infections are also fairly common. They're basically a dysbiosis of the microbiome in the ear that leads then to an overgrowth of bacteria, or it could also be fungi. And so this particular dog was first diagnosed with otitis way back in 2011. And so the next sort of bullet there is an excerpt of what was that first line of treatment. So typically when it's ear infections, you also have to do a long treatment course. And so this was treated daily for 3 months, and I highlighted in red the different antimicrobials. So 2 different antibiotics plus an antifungal and some steroid and painkillers. So this is what a dog got in its ear, both ears, for 3 months daily. I mean, think about how much, the selection pressure for resistance. But after the 3 months, the infection cleared, but then a year later, it came back. And it came back with a vengeance, and it was never able to clear properly. And so you had this chronic infection. And after 5 years, the poor owner decided that it was time to euthanize the dog, because the quality of life of a dog, if you have a dog with chronic otitis, it basically shakes its head continuously. And it's really a pathetic and sad sight. And so because they were about to euthanize the dog, the veterinarian thought, well, this could be a case in which we could test if we could use phages. Next slide, please. So basically, it's in Danish, but hopefully you will be able to make out at least the resistant profile. So they took some samples and they isolated, they did some microbiology and some susceptibility testing. And basically this was the resistance profile of the different bacteria that were isolated. And so you can see in one ear, you can have both a gram-negative, which is the Pseudomonas aeruginosa, as well as you can have the Staphylococcus pseudointermedius, which is a gram-positive, and have an incredibly also, varied resistant profile. And so to find just one antibiotic that will be able to target both the Pseudomonas and the Staphylococcus pseudointermedius was really almost, I guess it was impossible. And so when he contacted me, I hadn't been working with other than Staph pseudointermedius, I hadn't been working with any other phages, or let alone any other organism of phages against that as well. And he came to me with a Pseudomonas problem. So I went to the literature, as any good scientist does, and sort of looked at, well, what was being done around Pseudomonas and ear infections, and came up with 2 papers that looked at, were describing treatment or the use of phages to treat Pseudomonas, ear infections caused by Pseudomonas. And I've listed them there. The first one was using a monotherapy, so just one phage, and the subsequent one was a cocktail of 6 phages that they tested in dogs, but they also had a trial in humans, which was a phase 1/2 trial to see if you could treat chronic infections in humans. Next slide, please. So this led me to believe that, this was possible. It has been done, but in Denmark, we have no phage preparations for neither animal or human use. But at the time when I was looking at, what were people doing, I came across a paper in Belgium where they were doing a safety trial of a phage cocktail for Pseudomonas in burn victims. And so I contacted the author, who then put me in contact with one of the collaborators, and she was completely open to helping me and would give me any of the phages if they were worked against our strain. So I sent the Pseudomonas that we had isolated from Snoopy's ear to Belgium. They tested it against the cocktail, and only 1 phage out of the 6 was active against that. And we figured, well, they're going to put the dog down anyway. Why don't we go ahead with this experimental study with just 1 phage, knowing that we would not get it's not expected to have treatment success. And so this was approved by the Danish Animal Ethics Committee as a sort of a last resort treatment for chronic ear infection. And so we started with a 0.5 ml dose containing 10 to the 7 PFU of the phage in PBS. And this was administered directly into the ear, and then we massage the ear in order to make sure to get the phage to penetrate. So if you don't have a dog, or if you don't know what an ear infection looks like, especially one that is really bad, you have a lot of pus, you have a lot of debris, and so it's almost like a biofilm in the ear. So the access of a phage then to the organism is very, very difficult. So you have to do as much as you can to make sure that the phage comes in contact with the bacterium. And basically within a few days, we managed to see a log reduction of the Pseudomonas. But basically at the end of that trial, we never ever got to zero. We never completely cleared the infection from a bacteriologic perspective. But interestingly, from a clinical perspective, we had good clinical outcome in that when you have a chronic ear infection, you have ulceration in the ear. It's a very nasty infection. But what we were seeing was that the skin was regenerating, the wounds were healing, the dog was doing better. The owner said that the dog was happier, was more interested, could sleep because the head was not shaking, and really was much more lively. And so that turned out to be a very positive experience with phages that we had. Next slide, please. So this was just so the Pseudomonas story was a small story and was just by the way in terms of contacts. But moving on from my favorite organism, this is Staphylococcus pseudointermedius, and we knew that our temperate phages were failing. We still had this problem of what do we do with the MRSP infections. And so at the time, I was also looking at different, what was being published in terms of phage license, and came across Vincent Fachetti's work at the Rockefeller University in New York. And so together we applied for a postdoc project from the Danish Research Council that got funded. I was quite lucky, and I managed to get almost $500,000 for my postdoc, which at the time was my second postdoc. So phage license, essentially what a phage does, once it replicates within a bacterial host, it needs to escape the host. Angela, can you click? I think there's something's missing. Yep. And so it uses this two-component lysis system made up of an endolysin and a holland. And so hollands at the time, I mean, being very small hydrophobic proteins, you don't want to work with a hydrophobic protein. So the idea was that, well, and also we wanted to use this topically, and so endolysins just kind of blends itself to that. And these are basically cell wall degrading enzymes. I'm not going to tell you all because you're all phage people, and I'm sure you can read a lot about these phage license. Next slide, please. And so this is what the gram-positive lysin looks like. It's made up of these two domains. One is the catalytic domain, the bit that is going to chop up. Can you click again? Yes. So one that is going to chop up your cell wall, and where it cuts in the cell wall is dependent on the recognition or binding domain. And so you can have them attack various different parts of the cell wall. And the picture below is from Vincent's work, showing that when you treat with a phage lysin, is that you destabilize the cell wall, and then this causes, because of the difference in the osmotic pressure from inside the cell versus outside the cell, you would then have the phages, the cells popping. Next slide, please. So these, the term was then enzybiotics, and if you look in the literature, there'd been a number of good studies showing, very good results in vitro as well as in animal in vivo data, and people have shown that they work really well by itself, or if you use them, or they have a synergistic action when they're combined with different antibiotics. Click again, please. So Vincent and his former PhD student, they went on to form a company called Contrafect. And at the time, they were just about to launch a study on, or at least one of the license against Staph aureus was going to go into clinical trials. And it's called CF301 or exabase. which is supposed to be, click again, please. I don't know why we have all of this. Yes. So it completed phase 2 human clinical trials in early 2019. And it was given as an IV administration versus a placebo to see how well it worked. I haven't seen the data and I haven't heard, but if it's good, they're going into now phase 3, which I think is the only Lysin in a phase, at least completed phase 2 and probably waiting phase 3 trials. Next slide, please. So back to my favorite organism, right? So we now have a bacteria that is super resistant to almost, if not all, the antibiotics that you would typically use to treat pyoderma or skin infections. And it's because it's skin and ear infections, it's topical. When you get into systemic, it becomes a very different ball game. Topical applications tend to be a bit easier and controlled. So that was why everything kind of fell into place, that Lysins would just be perfect for us. And so we were working on 2 different Lysins during this project. Next slide, please, Angela. So the first one was called ASM2. And this what we got this from looking at the genome of our bacteria. And so it's an amidase, so it targets between the sugar moiety NAM and the peptide backbone. And that's where it's going to cut, that's where it acts in the cell wall. So we cloned it and we tried to express it, and we could see that we could express it. But the problem was always going to be, and this is the problem that is known with phages, is that it's really difficult to purify. Because for some reason they form very hydrophobic proteins and with very poor solubility. And so one of the ways around that, next slide, click. Okay, so one of the I'll get to the problem with the ASM2 one. And so the next phage Lysin that we were using was one that was isolated in Vincent's lab, which was a peptidase from streptococcal phage. And they had already done a number of work showing this broad lytic activity of this particular Lysin amongst a number of different bacterial species. And I was obviously interested in those that were the staphylococcal species. And it was very, I mean, they did a lot of work in terms of stability of the Lysin at different temperatures, as well as some basic in vivo work in a mice bacteremia model. So a lot of data sort of showing that it did really well in those setups. And I wanted to see if it would work with my organism. Next slide, please. So I took a trip. I got to stay in New York for a couple of weeks and go to the Rockefeller University. It's a beautiful place. You get to have beer with various Nobel Prize winners. So it's an incredibly enriching and inspiring place to be. And I really enjoyed my time there. And so I got to play around with this Lysin that they had there and testing it against my bacterial species that I was working with. And so like immediate, I mean, amazingly, it kills really, really quickly. And you get tremendous, I mean, it kills very efficiently. Next slide, please. And so brought the Lysin back and was doing a number of assays, because at very high concentrations, you can't use very high concentrations of an enzyme because it's very expensive to produce that. So I wanted to see how low I could go. And so tried sort of various titrations at different concentrations of the enzyme, what would be the killing effect of that. And really was able to show that even at 4 micrograms per milliliter was a very good killing efficacy. Next slide, please.
So Arshnee.
Yeah.
I can go through to your next slide.
Angela.
Coffee break. Coffee break. No, coffee break. Seriously.
There we go. Okay. So then I was trying like different things about if we changed if we looked at dosaging, not just giving one one dose, but that if we at time intervals gave different doses, what would be the effect of that? And really at a second dose, it was marginal, right? We got another log reduction, but it was not like typically very exciting. But still, I think it's quite amazing that within 2 hours, you can almost kill all the bacteria that you had, starting off at, at 10 to the 7, I guess we had 10 to the 8th, and then getting down to almost less than 1,000 cells. Next slide, please. So basically, as you said, we never actually solved the issue with the purification problem, and there are different ways of going around that in terms of you could do it in a gram-positive expression system, maybe because we were using a gram-negative system, that would in itself create problems. The other thing that people have been doing is by chopping up the enzyme. So basically, instead of using the entire open reading frame as it is, using different parts of it, focusing on the catalytic domain in the hope that then that recombinant protein, lysin, would be much better soluble. And in terms of PLI-SS2, I mean, since it did so well, we were looking at doing clinical efficacy trials in dogs, because the work was done in mice and we wanted to see it happen in a natural infection. We didn't want to create an infection because at the time we didn't have a good model for creating a skin infection. When you create skin infections, it's about taking you do it in a mice and you shave the mice and then you scratch the skin of the mice with a scalpel or with sandpaper to create this abrasion, and then you put your bacteria, and then you hope that it will then cause the infection. Well, it should cause the infection. It's been given all the right conditions. So we were looking at, I mean, to do it in Denmark with all the regulation that Denmark had, we thought we'd do it in the US at the University of Tennessee with a collaborator of ours. And we really wanted dogs that were presenting at their dermatology clinic for treatment. But I mean, it was also incredibly difficult because you couldn't, when you do an efficacy trial, you have to compare it against something. And because these are dogs that do have an active infection, you cannot do a placebo. So we had to, the setup would be that you would test the lysin against an antibiotic. That will be a one-on-one versus trial. The other thing was that, you couldn't deny patient care. Is that, well, what if your phage didn't work and you had, I mean, these animals they do suffer. How would you deal with that? And also to do a clinical trial in a dog was incredibly expensive, even in the US. So next slide. So we actually didn't get around to doing that. And so I have all of this data, and as well as all of this, the constructs and things like that sitting in my freezer back home in Copenhagen. But we've been discussing a lot about lysins and sort of the pros and cons of that, thinking of it really again from a therapeutic perspective. It has a number of pros in terms of rapid lysis. They've not been able to find resistance to it. You see synergy with antibiotics. It appears to be non-toxic from all of the safety trials. But the con is that it's kind of like a once-off. One enzyme, once it does its thing, it's done. Production costs are extremely high for an enzyme. And also, if you look at an infection, it's never very clean. You have a lot of other substances there to which the enzyme would bind to. And also, we didn't really know if there would be any immunogenicity. There had been some studies showing something, which would also neutralize the enzyme before it was able to do its business. So I think that, I think there's still a couple of kinks that need to be sorted out before this really becomes something that you can go down to your vet, to your pharmacy and buy. Next slide, please. So this leads me to another project that I was graciously asked to be a part of. It's called Antibiophage. It received funding from an EU funding agency, which is now defunct actually, but a project that received almost 1.5 million euros. And it was a project that was led by Robert Atterbury from the University of Copenhagen, Nottingham in the UK, with partners in Belgium, France, and then us. And so next slide, please. And I put all the names of the different people involved. Click. Okay. So the target of antibiophage was avian pathogenic E. coli, or short known as APEC. And this really typically causes, as Angela said earlier, it causes colibacillosis in poultry. And it can be, the infections can be really bad. And I put some ugly pictures there for you to see what type of infections they could cause. And looking at the apex strains that have been circulating, there are different serogroups, but the main or the dominant ones, at least that we see in Europe, is O1, belonging to O1, O2, and O78. And it's one of the leading causes of mortality and morbidity amongst poultry, in poultry production, and has an enormous impact on production in terms of reduced egg production, loss in body weight, and can also cause death of the animals. There is an E. coli vaccine called Pulvac, which is really against the O78 strains. And they've been looking at the efficacy of the Pulvac because it's very much dependent on when you give the vaccine will determine its efficacy. Or the other option is to use antibiotic therapy. But APEC overlap with human XPEC. So there have been studies showing similarity between isolates from urinary tract infections in humans and APEC from poultry. Next slide, please. Click, okay. And so how the project started was that we had a number of Apex, our partners, as well as some other partners who's kindly on the call today, Patrick, who at the time was also based in Belgium, sent us, so we got a collection of Apex strains trying to represent the dominant serotypes that we would find in Europe, as well as looking at different resistant profiles. And so at the end of these 122 strains, looking at resistant patterns and serogroups and all of that, we came down with a panel of 21 isolates. And we also included the Pulvac strain in here. And we also had 2 strains that were going to be our challenge strains in our in vivo models later. And so phages were, again, was from the collection, from various collections that people were gracious from in the consortium, and also Patrick shared with us. And so we were running parallel experiments. How do you whittle down 122 bacterial isolates and 100 phages? At the end, yes, we got 1.5 million, but that's not a lot of money. And so we decided to do sequencing of all the phages, using sort of genetic similarity between the phages because we're interested in genetically diverse phages. And then we did, as I said, we did a selection of the apex strains. And at the time we were doing this bioscreen. Bioscreen basically is an ELISA reader looking at growth, bacterial growth, and measures bacterial growth based on turbidity or OD readings. And so we thought that these were really nice, simple experiments we could do to avoid having to do all the traditional plating that real scientists do. And so we did a number of combinations of, with our phages and the different strains in order to, and then that's the curve that you see there. And then we got a very good friend of mine, a statistician, to help us to, define some parameters that would allow us to then select phages or not. And so that's the heat map that is on the far right. And so we're looking at isolates that had good host range, so was able to lyse many different bacterial strains, but did not lyse the Pulvac strain, so the one that was in the vaccine. And so at the end of this experiment, which took almost a year, we ended up with 13 phages, which we then confirmed by the more traditional plating. Next slide, please. And so came the next part is, well, how do you pick a cocktail? And so we try to be pragmatic in our approach. So we tested first dual combinations. So the 13 phages that we had, one of them was a Ciphroviridae and the 12 would belong into a Myoviridae. And so we did combinations with this where we kept the one that had the Ciphroviridae was the constant in all of the cocktails. And then we did 1 times 1, 1 times 2, and so forth. So at the end, we would have 12 combinations. And so we tested again in our bioscreen using the model strain, which is called CP6, which is an O1, belongs to O1, and quite a nasty strain because we know from our infection experiments it's very virulent. And so what you can see in the picture in the graph is that initially when you combine the bacteria with the phages is that you see slight growth and then suddenly it dips around 1 hour. And then in some cases, for some combinations, it continued to be at the flat line, which is 0. That means the wells are completely clear. There's no bacterial growth. But then at some point, you do have phage-resistant mutants, bacterial mutants arising, and that's what you see as growing in the subsequent growth curves. So we did look at if it was negative, and I'll show you some things. We would take bacteria, we will take an aliquot out and plate that just to confirm that, yes, indeed, you don't have any bacteria there. And yes, we can still isolate our phages. And where we saw phage-resistant mutants, a couple of them we were interested in because our partners in Belgium were looking at CRISPR. But we were also just interested to see, well, what was, what was the cause of these spontaneous mutants? And a lot of them were receptor binding mutants, because we could see that you would not get entry into the cell. Next slide, please. So then we tested those very same combinations against a second model strain. And this was the model strain that was isolated in France. And they were doing a lot of their experiments with this particular strain. And basically, those combinations just was really poor against this particular strain. And you could see growth in almost all the cases, really quite close to after 1 hour. And so that was a bit disappointing, but we continued anyway with our project. Next slide, please. So that was dual combinations now, and that's not the best. We wanted to get to having more phages in a cocktail, and so that came to triple combinations with our 13 phages, and essentially basically boiled down to 66 combinations that were tested with this phage 1, which is the Cipheridae, as the baseline, and then combinations of the other 12. And basically, this took many, many months, as you can imagine, testing these 60, and they were doing it in triplicate every experiment. And basically, this is just one combination of phages that we just call 137, and you can kind of see. The orange, the blue line is without phages, so that is the normal growth curve, and then the other ones are the different combinations of that, and where you sort of see, okay, you have a little bit of growth as we expected, and then it goes down, and then it's at 0 or close to 0, and then you have again the phage-resistant mutants arising. Next slide, please. But sometimes we were super lucky, and we saw this, and it was quite consistent. Out of the 66 combinations that we tested, 8 of them on our CP6 produced this kind of profile where it just consistently, and I think we repeated the combinations at least 6 times and verified with plating that this was the case. So we were really, really happy, sort of almost 2 years into the project, that we were getting these results, because it was an informed way, because how else were we going to choose what phages to put into our cocktail? Next slide, please. And so we took those 8 combinations, which we knew that the dual combinations were not working very well, but nonetheless, we tested it against the French, what we call the French model strain. And we didn't really see much difference in the sense that it didn't really work very well against this O2 that they had in France. Next slide, please. So we sent those 8 cocktails to our collaborator in France at INRA, Catherine, and she had set up a model and has a very nice paper out on these chicken embryonic lethality assays. And basically, it's been used as a model to assess bacterial virulence, and they have then adapted it to phage efficacy. And so they use these fertilized eggs and able to inoculate the eggs by inserting a very thin needle and being able to deposit the bacteria as well as the phages that we were using. And so it's a really nice model, because it's a type of in vivo model, but it's not quite the same as having a live animal. The downside is that you don't have the same kind of, you don't have the immune response, but you do have certain defenses in terms of, as part of the chicken amniotic fluid, they are sort of antibacterial agents in there, which would offer the defense as well. And you basically, once you infected, and when you look at lethality, you can hold it up to the light, this candle method, in order to see whether the embryo has survived or not. Excuse me. Next slide, please. And so I've tried to just color code, and she did it with her French model strain, which in our in vitro experiments did not do very well. And so she was looking at sort of embryo survival. And so I just color coded it, and she did that in comparison with an antibiotic treatment, just to have as something to compare our data to. And so in blue are the ones that did, so the ones in green are the ones that we, unfortunately, we saw some weird things. Like, it depended on which, I guess biology is the way it is that it worked really well in the presence of one phage. So if it was phage 1 plus our phage 4. And I can't tell you offhand what that phage is, but I'm sure it's in my notes somewhere. And basically the ones in the green was the ones that were the best combinations in terms of rescuing, or the embryos survived. And as I said, the con with this kind of approach is that you don't have the immune response, which would be quite important in a proper in vivo challenge experiment. But nonetheless, it was a way of sort of whittling down the cocktail that we would ultimately use in our in vivo experiments, because we simply didn't have the money and to have multiple experiments. And so those were so we took the 2 best cocktails from this experiment into our in vivo experiment. Next slide, please. So the in vivo experiment was a model, an infection model that was set up by Jens and Ida at the University of Copenhagen. And basically it's a salpingitis infection model where the birds go under, they open the birds, pull out the salpinx, inject with the bacteria and the phages, and put it back, and then close the bird up, and then you monitor the birds and look. And when they die, after you euthanize them, you can look at lesion scores and do a lot of microbiology. So as I said, we took the 2 cocktails that Catherine had selected as the best from the egg experiments, and we did the challenge with CP6, which is the model strain for this particular infection model. You should note that if you change the model, the challenge strains, you also change the dynamics of the infection. So we wanted to use something where we knew how this model strain is going to behave in our infection model. So this is how we set up the sort of the treatment groups as a group of birds that didn't get anything, bacteria or phage, one that only got the phages, one that only got the bacteria, and then one group, which was our treatment group, that would get the bacteria and phages at the almost simultaneously. First we put the bacteria, and then we put the phages in. But that we injected it along different parts of the salpinx lumen. And so after 48 hours, the birds that survived, that were doing well. Of course, if the birds were not, they were monitored, and if they were not faring very well, for example, in the group 3, they were killed. And so we did dissection, we looked at pathology, and then we took samples for microbiology. And this was from the ovary itself, blood. We took mucus from the salpinx, because in a salpingitis, you have a lot of mucus production in the salpinx. And so we scraped the salpinx in order to get this mucus. And then we also looked at the spleen. Next slide, please. And really what I wanted to show you here, I'm not going to show you all of the details of that because we're currently drafting our manuscript, but basically phage cocktail 1 didn't work at all. So if we look at the graph, the very first one is the control, or the negative control, where there's nothing. And therefore you shouldn't see any lesions. The second one would be the one that only received the phages. Again, no lesions. The third one is the one that received the bacteria only. So there you'll see a lot of different lesions. And the last one is the treatment, the one with the phage as well as the bacteria. So cocktail number 1 didn't work at all. It failed. I mean, you can look at the lesion scores are almost comparable with that of the one that didn't get treated. But cocktail 2 did very, very well. In fact, it managed to rescue the birds. In terms of lesion scores, we saw almost the same as if the birds were not infected. Very few lesions, and clinically they looked very well. Yeah, so that was basically the moral of this story is that almost 1.4 million euros later and almost 3.5 years of work, we come and 100 phages, 122 apex strains, we come up with one cocktail that seemed to work well in at least an experimental model. How it fares in the field remains to be another discussion. And also, I think there's a number of still open questions is how you would administer it, how often will you give it, in what format will you give it, more than that, what are the production costs? I mean, it took, just for this experiment, we had to produce a cocktail that was very clean and, rid of exotoxins and very high titers, which also took a while to do that just for this little experiment. If in terms of long, so bigger production, remains to be seen. One of the things we're looking at is the phage-resistant mutants, if there were any, and that's kind of work that has been done by our Belgian collaborators, looking at CRISPR and whether we would see anything in that. And unfortunately, I don't have that data to share with you today. Last slide, please. Next slide, please. And really, I could never do all of this work myself, and I'm very proud to have very many people that worked with me. I would say, even though I supervised them, this was them really doing a lot of the work. I have a fantastic postdoc who now works at a biotech company looking at utilizing phages for therapeutics. Several master students, great technicians, and also been very fortunate to be involved in PhD projects with Patrick and Liz Wellington at the University of Warwick. Next slide, please. And really, I think the moral of this story for somebody who's not a phage expert, I couldn't have done this without aligning myself with very good phage people. And this is people at the University of Copenhagen, in Warwick, at Ghent, at Ross, at the Rockefeller, Nottingham, in Belgium at KU Leuven, as well as in INRA. So the moral is to find good people to work with. You get to have some fun. But in terms of where we ended up with anything that I could go to the pharmacy and buy, I think we're still far away from that. And I've been having a number of discussions with Zoetis and various other people who we have links to as part of other projects. And really the regulatory frameworks are just not there in order to enable phages to be approved for use in the traditional, it would go to the same rigorous program or cycle as an antibiotic, with a new antibiotic, which is almost 10 years, and sometimes you can invest almost $1 billion in all of those clinical trials. And I think that's my last slide, and I just wanted to say thank you.
All right, thanks, Arshnee. Now I have to go back into the original screen and look at all the questions. I hope they're not too many.
Unless people want to put your hand up and then you can unmute.
Yeah, I don't know, Jessica, do you know if people can put their hand up and then I can just do one by one and then they can ask, and if they want to have their video on?
Yep, that should be fine. People can find the actions button and use the put your hand up, or where is it?
It's under reactions.
Okay.
At least it should be. Or you can just unmute yourself.
Yeah, right now we have it so that you can only unmute if I specifically indicate. So you have to raise your hand.
Okay, maybe I'll start asking the questions. And Adamu was asking, was therapy on otitis externa using phage or by spray or injectable?
So basically we kind of dripped it into the ear and then we massaged the ear. Maybe I can just, So basically we just took a syringe and we sucked it up and put it into the ear, dripped it, and then you massage it in order for it to get, to go down, and using gravity because the dog would be sleeping.
All right. And Sandra is asking, what do you recommend, phage cocktails or just their enzymes?
I think that's a bit too generic. It's very much dependent on what is the infection, how are you going to administer it. So I think they both have pros and cons. So I would not say one over the other.
Right. And Meshach is asking, can lysogenic phages be engineered to deliver phages straight into antibiotic resistant bacterial cells? But I think we'll be ending up with GMO problem here.
Exactly. Anything where you genetically modify it, it becomes treated as a genetically modified entity, organism. And so the regulation is even more stricter on that.
Yeah, another question from Joseph is, why did you combine myoviruses with cephaloviruses?
At the time, the sort of advice was that because they would behave differently, and that it's better to have phages belonging to different families in terms of, I don't know, they just said it would be better. As I said, I'm not the expert, so I wouldn't know.
Yeah. And Varun is asking, were these phages in a cocktail of 3? Were they randomly chosen? And what were the basis of choosing?
So the phages, I assume you're talking about the Antibiophage project. Yeah. And so the final phages were 3 different phages that were in a cocktail. And when the process we took was sort of an elimination process, trying to do everything we possibly can to get what we thought in vitro would be the best cocktail, in the in ovo model would be the best one, and then that would be like give us all the confidence that this would be the one that would work in the in vivo model, which didn't work out like that. So we tried to do this very pragmatic, stepwise kind of elimination to get to making informed decisions, not just saying, well, I'm just going to take whatever I want, it's randomly based on our host range and put them together. That's not what we wanted to do.
Yeah. However, we're back into the Mayo and Cephaloviridea question. And was the outcome, how would you know the outcome was from myoviruses or with the cephaloviruses?
So we didn't look at, so in the in vivo experiment, I know that they tried to look at trying to isolate what would be the most dominant phage that we would get. And I don't really remember the data if they were able to, because I think for one of the cases, one of the combinations, one phage had 2 different strains, or at least 2 phages would be able to block on the same strain. So it would have been very difficult to figure out that this was this one, you know, sort of the relative ratio of the different phages.
All right, Francesco is asking, the phages used in your combination, do they have the same receptor for binding?
We didn't get into any of that. So again, this is what I mean is that I would never call myself a phage biologist, because I think a phage biologist is really interested in the biology of the phage and understanding all of those bits. And I only see them quite arrogantly or ignorantly as a tool. And I'm sure the real phage people would be looking into all of those things, and we didn't look at that.
Yeah. Sabina Green has asked, have you tested your APEC phages on XPEC phages from humans?
XPEC strains, rather.
Yeah, but from human beings.
Yeah, no, we didn't.
Okay. Okay, so Newton is saying, great learning curve, doctor. How is South Africa community and how your knowledge will benefit the African researchers based on your learning curve?
I think there's a lot of promise, but I think there's still a huge gap there. And I think people need to understand what it takes in order to what you do in your laboratory to what happens when you have to actually go into a shop and buy it. It's important that you look and understand that process, because that process is incredibly difficult and long. And I know in our grant applications, we write how we're going to save the world with this project and we're going to, you know, tackle. But the reality is that we're far from that. And so that doesn't mean we shouldn't work at it. I think we still lack a lot of field data to show that this works and what works under what conditions. And will it work in one country? If I use it in another country, is that going to work? Because one also has to understand from big pharma is that they will not invest, because ultimately they buy the product from you and then develop it, because you don't do the development, at least I don't, not in my research institutions. And so they need to be able to invest and see an economic investment in that, because the investment that they put in is almost $1 billion. And so they have to recuperate that. And so if you don't know how it's going to perform in terms of resistance development, efficacy, there's still so much that we need to figure out.
Yeah. So YD Soleil is asking, I want to believe the lysin that you're talking about is a phage lysin.
Yes.
Is he correct, or is it a bacterial lysin?
So it's called an endolysin.
Yeah, but still. Yeah.
So, well, it kills bacteria, right? It doesn't kill phages.
True. But
So,
a bacteria can encode it based on lysogens.
true.
I
Yes,
think that's what he's trying to say.
I mean, they're derived. So prophages might have these endolysins with them, but when a prophage is in the genome, it's dormant, right? For the most sense, it's repressed,
Yeah.
and it's only when that repression comes out, and then you will have the activation of the endolysin. So I guess I'm not really understanding the question.
Maybe he'll ask again. So I'm saying excellent talk. Tesfaye is saying excellent talk. Gavin, thanks, great talk. I'm just giving you what people are saying on the chat. Golshan, great talk. One of the best presentations. Martin, great presentation. Adam, brilliant work, Arshnee. It looks like a very good start to develop a treatment ready for production. Adam, I won't even pronounce the other one. So YD again is back. How did you get the lysin ASM2 out of the phage?
Basically, because I had the genome sequence, I could see I designed primers on either side of the lysin and was able to amplify it. But I used the cloning system that allowed me to be able to amplify the open reading frame and then be able to insert it into my vector.
Good. Ivy, interesting, great work. Okay, so Tesfaye is saying you attempted to isolate from dog pups, but what was the worry to look for virulent phages from the environment, like sewers? They're abundant there, so why didn't you look into the sewers?
So we didn't find any. So I mean, we assume that, I mean, poop is a good enough area. And also you do have, you can find Staphylococcus pseudointermedius in the intestinal tract, like Staph aureus. So we thought it would be a very rich thing. We also tried taking it from nasal secretions. We're taking it from the environment. We couldn't. It's so difficult to isolate these phages against Staph pseudointermedius.
Yeah, so Varun is saying, great work, Arshnee, enjoyed your talk and thank you. So Adamu is saying, well done, Arshnee, interesting, educative phage work. What did colibacillosis, we did colibacillosis in Malaysia, however, our myovirus is single phage with interesting, share your email, please.
I'll put my email in the chat so you can have it.
Yeah.
Whoever wants to email me. Yep.
Okay, so Alison Lowe says, like, your pseudophages turned out to be lysogenic. What do you think lytic phages exist for pseudo?
Do I think that they exist? I hope so. I mean, I had an entire PhD student that spent 3 years trying to hunt for them and ended up not actually finding them, which was very sad for her, for Muriel. So I don't know if it's in relation to the isolation protocol. Maybe it requires a special isolation protocol. I don't know. But we tried as best as we could and we couldn't. And I would hope that they're there. But as I said, I don't know enough about sort of phage biology to understand if this is a common phenomenon, if this is seen for other types of phages against other bacterial species.
Yeah. So Priyanka is asking, like, minimum, how many phages do you think are required for doing a phage therapy?
I think when you look at the literature, they say up to 6 different phages, I think is good. Anything more than that, it gets a bit more complicated, I think, in terms of dissecting, you know, how it's going to work and things like that. But we only went up to 3 because I don't know, actually, we could have, I think we did try, you know, with 4 phages, but we saw the same kind of results. So would it be better to have 4? Would it be better to have 3? I don't know.
That I can honestly say, I think it depends on production. And in our current project, on the phage project with salmonella, the maximum they said we can have, and we're trying to create a product that would be very suitable for their African setup. So they just said maximum would be 6, but then we'd have to incur costs on production, shipment, but shipment would mean we're shipping powder. But by the time we're making phages in a powder form, that's a bit of a different ball game. And then we need to also understand that the efficacy might not be the same.
Exactly.
Yeah.
I think there's still a lot of applied work that is missing, sort of the next steps, because we get the funding for the very basic stuff. We don't get the funding for the continuation in the next phase.
Yeah. And then Tesfaye is saying, I remember the benefit of using divalent cations added to the media, like in soft agar for Staph aureus, unlike gram-negative phages. Do you agree? I don't agree.
I'd say I don't know.
No, because we also add cations, and we have gram-negative phages, just because you have to understand, maybe with penetration or binding, it's like a game of Lego. You might need like little tiny thing to add up to something that will allow the penetration.
Absolutely.
Yes, but I also have a question for you. So when you say the phage did not penetrate, did it mean that it did not bind or did not inject?
It didn't adhere.
Okay, all right.
Yeah.
All right, I hope I have a last question. What is the limitation of the phage approach? No, in your own.
In my, from my experience, I think the issue is production, and the issue would then incur on stability and how would we administer it. And I think we still don't know what we should be doing. When you say a phage for therapy, it's a different ball game in terms of regulation and things like that. I didn't show a project that we're doing with streptococci. We did with streptococci and mastitis in farms. So Streptococcus ubris is it's considered environmental mastitis. So it's usually seasonal, and it's dependent on the presence of the organism in the environment. And typically it's on the bedding. So when the cow sits down, that's when you get the contact with the udder, and then you have the infection. So we were looking at, so one of the things we thought, it's like, we don't say therapy, because therapy is a no-go and requires a very different way of thinking about it. So we thought, okay, let's call it disinfection. And one of the challenges, so I managed to get some phages. Actually, it's a very interesting project, which unfortunately I had to stop it because I was coming to Kenya, was that when we sequence the phages, they don't look like anything in the database. So I still want to go back, or at least get the phages here to be able to look a bit more as to what that would look like. But one of the things, I think it's important to think about the infection that we're trying to deal with or try to solve, is that we thought, okay, we could just spray the phages, in the environment, and then it would, clean everything up. But what you don't want is moisture. Moisture is the thing that could cause the bacteria to proliferate. And so you want to keep the environment as dry as possible. So the thinking is about how are you going to administer the phages so that you don't actually put it you don't create conditions that exacerbate the condition, because delivery, I think, is incredibly important, plus all the other work that needs to come in terms of dosaging and what MOI, not MOU, do you need, and things like that. I mean, we choose fixed amounts, but is that better? Should we be trying lots of other ones? What is the magic number? I think we don't really have that.
Yeah, but for example, for people who want to go into wound treatments, whether it's with human, with animals, and now there's this further dynamics playing in, and you treated a dog infection and just had to massage it a bit. What would you think would be the bigger play of phages in wound infection?
So I think if you want to go into an area, you should understand what is the current treatment protocols and why. And so you also have to understand the disease progression, understand what is the bacteria doing and why is the treatment like this in order to prevent that. So as you and I have discussed, Angela, about wound treatment, it's a similar kind of thing, right? You typically want to dry out the infection. Any moisture in that area will cause the bacteria to proliferate. So I think and the other thing is about penetration. is that one thing is that, you know, the skin is multiple layers, and to be able to penetrate, that was also the same thing with the phages that came up, as well as the Lysins, is how you're going to get very deep into the wound, considering you will have pus and all kinds of other material kind of debris that will steal or hide your bacteria. So you need to really think about all of those things of how you're going to get access, how is the bacteria that you want to target, what is the access. The other thing with wounds is that, is it one bacteria, or is it usually polymicrobial, the infection? So which one are you going to target? And are you going for zero, like killing all the bacteria? Or do you want to do it where you can get a little bit, and then you want to give time for the host immune system to kick in and start, you know, getting the process? And that's what we saw with the otitis, was that we never ever got to zero, but you get it low enough in order for the skin to regenerate. So you give almost like a break then for the host to then start repairing itself. So I think when you want to get into a field, you really need to do your homework and really understand.
Yeah, but what do you believe about synergistic? Now you're AMR. Could we use phages with antibiotics, but at a lower dose of the antibiotics? What do you think about it?
So the AMR lead, I would be a little skeptical, right? Because considering using low dose, unless you kill the bacteria and you do not create resistance or promote resistance in other flora, because we never work with a single. And that's the problem also when we work in the lab, is that we work with one bacteria, a nice clean environment, everything very perfect. But the reality is that you have a lot of bacteria there when you want to treat an animal or anything like that. And those dynamics are really important as well. And so when you want to treat synergistically, I think the reservation.
Is she frozen for you, Angela?
I think so. Yes. I think I'll tell her, Arshnee, because I can hear her talking.
Oh.
Yeah, and maybe she's continuing like explaining herself. Okay, I'll just, I hope she's not frozen. Hi, Arshnee. No, she's not back yet.
Not our best technical day.
I know. Coffee breaks.
Yeah, so much coffee today.
Maybe as she comes back, I could share my screen just as a conclusion, and then she'll come back and explain her point. All right. I think she's frozen.
Are you able to go get her physically, or?
Yes, she's just like down the hall, like just 2 minutes. Yeah, she's here. All right, actually, you're frozen. You're frozen. Come. I don't know whether we need to wear masks just for. We'll sit far away from each other. So we... Okay, I'll point my computer. All right. Right. Okay. So we just lost you at some point.
Yeah, my internet died in the office.
Oh, just, okay.
I guess we're done.
No. I'm not leaving the meeting.
Stop sharing.
Okay. So we were talking about the synergy, and yes, and a lot of people are coming in and asking you, phage and antibiotic combination, is it the way to go?
I think if you can show that the use of the antibiotic at a lower concentration or at any concentration will not select for resistance in a commensal bacteria, then yes, why not? I mean.
Yeah. And do you think the bacteria will develop a resistance to phage therapy, or did you record any phage resistant strains in your previous experience?
I mean, you could see in the bioscreen is that you see in some cases you saw within an hour, you had bacterial growth, showing that you do have these spontaneous mutants that arise incredibly quickly. What does that mean in terms of therapy in a real life person? I think it also happens too. And we saw that in our birds. We were able to isolate the bacteria. So I think that is the battle, right? In terms of resistance, and how quickly does your bacteria develop resistance will determine the success of your cocktail. And isn't that the premise of adding so many phages to your cocktail? Whether, bacteria evolve, things, everything evolves, right?
Yeah. How do you approach naming a phage isolate?
So, Rob,
have you had any to name any?
Yes.
I should
Rob
say,
Levine and co have. No, I wish I couldn't. I'm not that arrogant to name them after me. So there is a very nice paper showing you how to do that. There's a way of naming phages.
Yeah. And can we use lysogenic phages to deliver antibiotics into MDR bacteria?
So not really, because an antibiotic is a compound.
Maybe I think he's saying about the CRISPRs to digest antibiotics.
CRISPR to digest the antibiotic. I mean, CRISPR only identifies, so it would need to identify some sort of
module
protein signature in the antibiotic in order to degrade it. But you don't want to degrade the antibiotic.
So how did you preserve your phages?
We didn't actually. We just kept them in acid buffer in the fridge, and they seem to work fine for many, many years.
All right. Okay, I think we should be done now. So I'll just share my screen.
Thank you for having me, everybody. It was really, really fun.
Yes.
I think I will go home now.
Yeah, but we have a few events coming up. So I'll just share my screen. Does anybody hear me or?
Yeah, I hear you. I see your screen.
Yes. So courtesy of Rob Levine, he got to me today and he said, we have conferences coming up in 2022, 2023, and 2024. And I'm hoping in the near future, we'll hold it in Africa. But he said, please be a member. It's free. And you have advantages of being part of the ISVM network. You can contribute and develop new ISVM projects. You're eligible to serve as an ISVM officer, and then you can have endorsements and promotions of related initiatives. And then you can increase visibility and recognition to your science. And then we also have poster prizes at the Viruses at Microbes meeting. So please, 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 please sign up. And then, as we know, the VOM was postponed to next year. So hopefully we will meet there. And please don't forget to register. And thank you for all your time and attention. And I don't know if Olafum is still here to show. I think we've had a very, very long meeting.
Very, very long meeting.
Yes, but please share the last slide and then we can close it for the day. I'll stop sharing. There you go.
Please, can you say this?
Yes, and we're so hungry right now.
Okay, so these are meals in Nigeria. From the northern part of Nigeria, we have a meal that is known as gote. Gote is made up of vegetable fonio, and you can also add a number of beef, chicken as desire. Very, very nutritious, contains balanced diet. This is from the south of Nigeria. We call it ekpang and kunkwo. It is made with cocoyam, cow leg, cow head. So rich. In fact, I'm also salivating because I know it's so rich. And we also have another meal, abacha salad. It's from the eastern part of Nigeria. It is made from cassava. shredded cassava, added palm oil, and you can also serve it with fish. And from the western part of Nigeria, we have a meal that is known as amala, begiri, and ewedu soup. It's with big meat. The begiri soup is made from beans. The amala is made from yam flour, and the ewedu is vegetable. It's very nutritious. All these meals, when you come to Nigeria, you can have a taste of it. We have some festivals that are also in Nigeria. We have the Arugungu Fishing Festival in the northwestern part of Nigeria. This festival has to do with the northern. All fishermen come around to Kebbi State, and they will now look for the one that will get the biggest fish in the river and will be given an award. We also have the Calabar Carnival. The Calabar Carnival is a carnival that is known as the biggest street party in Africa, in West Africa. Biggest street party. It's a very fun fair, different attires, very attractive. It is mostly done in December period. And we also have the New Yam Festival. The New Yam Festival is done in the eastern part. You cannot take a bite of the yam except it is being dedicated to the gods. So until this is done, that is when the community are now encouraged to eat the yam. It is done in the King's Palace with a lot of fun, different cultural dressing. We also have another one in the western part of Nigeria, the Lagos State. In Lagos State, we have the Eyo. This is a masquerade that attracts terror. It attracts a lot of people to the country whenever it is done. Very beautiful. The site is very attractive. So these are just few numbers of festivals in Nigeria. We have lots and lots more of them. We hope one of these days that we hold our Phage conferences in Nigeria, and I know that we all enjoy our stay. Thank you very much for giving me this privilege to present these few slides. Thank you. Over to you.
Thanks so much, Olufan. Jessica, do you have anything to say?
No, thank you everyone for coming, and thanks for organizing, Angela. This is such a great session as always. So I'm just glad everyone could make it, and yeah, nothing much to add.
Yeah, me too. And thanks everyone for coming through.
Yes.
All right, we'll see the people. I hope we'll see you the next time.
For sure. Yeah. Okay. Well, have a great night. Have a great
You
day
too.
for those on the morning side of the world, like me.
Or those who are going to sleep like me.
Yeah. Good night. Bye.
Good night. Bye.
Thank you.